Hello students, my name is Boniface, a biology teacher. Welcome to this presentation. I'm happy to be with you this day so that we can share some interesting concepts in reproduction. But as you all know, the topic of reproduction has a lot of things to cover, and we cannot cover all of it. Therefore, I have selected some few concepts which seems to be difficult to most of you students to be covered during this presentation. But why few concepts and not all? It's because the topic of reproduction has appeared to be difficult to most of the students. In such a way, they face difficulty to respond to some questions that comes from this topic. Therefore, I expect that at the end of this presentation, students will be able to answer some questions that will be asked from different areas of this topic of reproduction. But before I start, let me take you to some very important concepts that I expect to cover during my presentation. Here is a concept map showing some important concepts to be covered in different presentations I'll be making here before you. As you can see here, the topic of presentation is reproduction, and the subtopic is reproduction in plants and reproduction in human being. In reproduction in plants, I expect to cover structure of the flower, the concept of gametogenesis, very little pollination, and fertilization in a flowering plant. To put more emphasis, double fertilization. But reproduction in human being, later on after covering this part, we'll see the reproductive systems, the gametogenesis, fertilization, and birth processes. These are things which I expect to cover all the time I'll be with you. Dear students, what I expect to achieve at the end of this presentation is that I expect students to be able to explain the structure of the flower and the concept of Gametogenesis. Therefore, to start with, in my presentation, I expect to present the concept of flower. The concept of flower. Here, I'll have to cover structure of the flower, and also the function of different parts. After that, another concept that I expect to present today is the concept of gametogenesis. In gametogenesis, I expect to cover the concept of Microspore Genesis. and Megaspore Genesis. But in order for us to be able to cover these concepts, my presentation will be guided with some questions. There will be questions that will guide us to discuss these concepts and which will touch different areas of structure of the flower and the gametogenesis. To start with the flower. A flower is a plant reproductive organ. Therefore, whenever you see flowers, mind you that those are organs for plant reproduction. Therefore, when you cut a flower for your interest, it means you are affecting reproduction in plants. But to remind you, what are different parts of the flower? This is the question that you need to ask yourself. If you remember, in the topic of reproduction, I'm sure you learned that a flower consists of 4 main parts. The first part, which forms the outermost part, is the calyx. The calyces. This is the collection of several sepals. The sepals are green in color. They are green in color, and for that case, they are just for protection. Of the flower before it open. A collection of several sepals form what we call calyces. The second is a corolla. Corolla is a collection of several petals. Several petals form what we call corolla, just inside the calyces. These are colorful part of the flower. They appear in either yellow, purple, blue, etc., etc. Therefore, if you are just walking around and you happen to see a flower, the part that will attract you with its attracting color is known as the corolla. But also, inside the flower, there is another part known as stamens. The stamens form the male reproductive part. This consists of anther and the filament. The filaments. The collection of several stamens is known as androecium. It is this part where the male gametes are formed. Responsible for male gamete formation. The last part is known as the carpel. Which consists of stigma, the style, and the ovary. This forms the female productive part. Where the female gametes are formed. Formation of female gametes. But also the part where fertilization takes place. And lastly, the place where a zygote develops into embryo. To mention few, therefore, you see these are different parts of the flower. Let me take you to a sketch of a hibiscus flower showing these parts I have just mentioned right here. The hibiscus flower has a stigma. But here there is ovary. Then there is what we call the stamen tube. This is just the sketch showing the hibiscus. If you label here, these are stigma. And here we have the style. These are the anthers. And these are the filament. Collection of these forms stamens. But here we have petal. If you combine this and this, you get several petals. And here we have the sepal. If you combine this and this, you get sepals. But this is the epicalyx. This is just the sketch showing the different parts of the hibiscus flower. But the hibiscus flower has a unique structure known as a stamen tube. Also known as a staminal tube. A tube on which the stamens grow outward. Therefore, this is just a sketch showing the hibiscus flower. Dear students, as I told you before, During the beginning of this presentation, I said my presentation will be guided with some questions. And I have done with the first part of my presentation, which was concerned with the structure of the flower. Now, let us see some guiding questions from the structure of the flower. The first question here says the flower consists of the primary and the secondary parts. How are the primary parts modified for reproduction? This question requires to identify primary part and the secondary parts and explain how the primary parts are modified for its function. In my presentation, I just told you different parts, and it were 4 parts. But this question gives us another concept, that there are primary part and the secondary part. Let us go. Primary parts are those parts which are responsible for reproduction. They take part directly into reproduction. And these consist of the stamens, where the stamens consist of the anthers and the filament. A collection of these forming the stamens. But also there is the carpel, which consists of stigma, style, and the ovary. These forms the primary parts of the flower. But also the question required to identify secondary part. What is needed here is to understand the secondary part of the flower. These are parts which just support the flower, and they don't take part directly into the process of reproduction. It means here it consists of corolla, the petals, and the calyx, which form sepals. These are just there to support the flower to accomplish its reproductive function. For instance, I said the petals are brightly colored for attracting insects. But also in insect-pollinated flower, they do produce nectaries. In insect-pollinated flower, petals produce attracting smell. We say they are scented. For that case, they can support the flower by attracting the insect, that they will bring pollen grains. The sepals just protect the flower. For that case, you have seen the primary parts and the secondary parts. But since our question requires to explain how the primary parts are modified for its function, which is reproduction, therefore you'll have to explain something about the anthers. For instance, the anthers contain pollen sac. Each pollen sac has numerous pollen mother cell. Which gave rise to male gamete by meiosis. But the filament just support the anthers. Coming to carpel, there is stigma. The top of the stigma is sticky to allow the pollen grain to tuck in when it is brought by either wind or insect. Then there is a style. The style has a space in between to allow pollen tube to grow toward the ovule. And then there is ovary. The ovary contain ovules within which there are megaspore mother cells. The ovule contain megaspore mother cells, which give rise to female gamete by meiosis. Dear students, by having explained how different parts of the flower, particularly the primary parts, as I've said, modified for its function, you'll have responded correctly to this question. Let me now take you to next question. Question number 2 reads, draw a labeled diagram of a hibiscus flower with all accessory parts and stamens removed to show the female productive structures only. What are the accessory parts? The accessory parts are sepals and petals. These are accessory parts of any flower. Stamens are the anthers and the filament. Therefore, the question requires you to draw the remaining part, which is the female productive parts. Let us show you this sketch. These are stigmas. And here we have the stamen tube, the petals, the sepals, and the calyx. And here there are anthers supported with filaments. If stamens are removed, it means you will have to remove the stamen with its staminal tube, and you have to remove these accessory parts. And draw the remaining part. After removing those parts, the remaining part will be the stigma the style, and the ovary, which show the female reproductive parts. But if you remove all those parts, this is what will remain. I have done with question number 1 and question number 2. In the beginning of my presentation, I told you that my presentation will have 2 main parts. The first part being the structure of the flower, and the second part being gametogenesis. This is the process by which the gametes are formed. In flowering plants, there are 2 types of spores. Flowering plants produce 2 types of spore: the microspore and the megaspore. The microspore is formed in the anthers, and the megaspore is formed in the ovule. But what you need to understand is that the microspore is what will give rise to male gamete, and the megaspore is what will give rise to female gamete. Dear students, if this is the case, let us see how microspore formation takes place and how megaspore formation takes place. Before we go into detail, let us consider the microspore. The microspore is made up of 2 words. There is micro, which means something very small, and also there is spore, a small reproductive unit of either plant or even the protozoa. Also, there is megaspore. The mega means big. It means the megaspore is bigger than the microspore. The spore means a small reproductive unit. For that case, the formation of these 2 different spores is referred to as microsporogenesis for the formation of microspore, and megasporogenesis for the formation of megaspore. But in the beginning, I told you that my presentation will be guided with the questions. Let us see now different questions that can guide us into discussion of these concepts, the microspore and the megasporogenesis. The next question that leads us into gametogenesis in flowering plants is question number 3, which reads as follows: with the help of diagram, describe the process of microsporogenesis. Dear students, this question, as you have read, requires a description on how microsporogenesis occurs. To answer this question, you have to consider the following. First is the site where the process occurs. As you understand, this process occurs in the answers. The answer is the site where male gamete formation occurs. Each answer has 4 pollen sacs. In each pollen sac, there are many microspore mother cells. Mother cells, also known as pollen mother cell. Dear students, these microspore mother cells contain a diploid number of chromosomes of the parent cell. Therefore, the Megaspore Genesis involves 2 cell divisions. There is meiosis and mitosis. How this process occurs? You start with microspore mother cell. Which contain diploid number of chromosomes. These undergo meiotic division. And this is meiosis 1, giving rise to 2 haploid cells. Microspore. These are 2 haploid cells. Each haploid cell undergo another meiosis, giving rise to another 2 microspores. It means this is the second meiosis, producing 4 haploid cells, microspores. These are not gametes. They are just cells having haploid number of chromosomes. After that, each cell secretes the wall around itself. And get separated from each other. After that, each microspore grows to increase in size. producing large microspore. When this process has taken place, the nucleus in each microspore undergo mitosis. Producing two nuclei, of which the first nucleus is known as generative nucleus. And the second being the tube nucleus. Also known as vegetative nuclei. Therefore, a complete pollen grain contains the exine, the cytoplasm, the generative nucleus, and the tube nucleus. If you draw by making a cross section of it, you get a microspore which appear as follow. You have the exine forming the outermost waterproof layer, but also the tube nucleus, the generative nucleus, the cytoplasm, up to that, you will have responded to this question that will require you to explain how microsporogenesis occurs. This is a microspore that has exine, tube nucleus, generative nucleus, and a cytoplasm. However, the male gametes are not yet formed. Male gametes are not yet formed. How the male gametes formation occur? After the formation of these generative nucleus, the generative nucleus will undergo another mitotic division. Producing two haploid cells. And these haploid cells are the male gametes. But this process normally occurs when the pollen grain has grown into pollen tube. It means when it has landed on the stigma, absorbs nutrients and water, then give rise to pollen tube. The pollen tube will contain the generative nucleus, which will divide mitotically to produce two haploid cells, forming the male gamete. But because our question asks us to describe the process of microsporogenesis, the microspore, the question will end up here, where we'll have formed the pollen grain. And that will mark the end of this description regarding to what you were asked to do. Question number 4 reads as follows. With the help of diagram, describe the process of Megaspore Genesis. Here, the question requires to describe the process of Megaspore Genesis with the help of diagram. Where does Megaspore Genesis takes place? I said it occurs in the ovule. Each ovule contains the megaspore mother cell. These are diploid. Each megaspore undergo meiosis, producing 2 megaspore. Later on, each megaspore undergo meiosis 2, producing another 2 haploid megaspore. But out of them, 3 degenerates and remain with 1. The remaining 1 grows into a bit large megaspore. A bit large megaspore. From there, its nucleus undergo 3 consecutive mitosis. It means there is mitosis. The first mitosis produces 2 cells. Second mitosis produces 4 haploid cells. And the third mitosis produces another 2 haploid cells from each haploid cells, making a total of 8 haploid cells. These cells undergo cell rearrangement. In such a way that 3 haploid cells moves at the top, forming the antipodal cell. Two haploid cells remain at the center, forming the polar nuclei. And the last 3 remain at the bottom, whereas 2 out of them form the synergid. And the one of it form the egg cells, egg cell. If you sum up, you get 80 haploid cell. The resulting structure formed from this process is therefore known as embryo sac, with 80 haploid cells. And the embryo sac can be drawn as follows. Here, there is large egg cell supported by synergid. These two forming the synergid. two cells that remain at the center fuse together to form the diploid nuclei. Or each cell is a polar nuclei. But the three remaining, which appear at the top, form the antipodal cell. This is an embryo sac. Therefore, if you want to respond to this question correctly, you will have to go directly into stages from the megaspore mother cell in the ovule, how it is transformed to 4 megaspores, 3 get generated, remain with the 1, and the remaining one grows to increase in size, then undergo mitotic division 3 times, producing 80 cells, of which the 8 cells will be arranged in this order. The egg cell being at the top, supported by the 2 synagids, and 2 cells remain at the center, known as polar nuclei, which fuse to form a diploid nucleus, and also there will be 3 at the top. Dear students, I have done it with the question number 4. Let us now see other questions that can be asked in different ways, but from the same concepts. Question number 5 reads as follows: if the number of chromosomes in a flower X is 22, calculate the number of chromosomes in each of the following cells or nuclei and give reason for your answer. A is antipodal cell, B egg cell, C megaspore mother cell, D the polar nuclei. Remember, when I presented the Megaspore Genesis, I said the megaspore mother cells contain diploid number of chromosomes of the parent cell. Assume these were 20 number of chromosomes. After undergoing meiosis, it means these 20 chromosomes will have been reduced to half, which equals to 10. Then from there, the megaspore did not undergo any meiotic division. Rather, it went through mitosis. And the mitosis does not reduce the number of chromosomes. Rather, it restores the number of what? Of chromosome. It means from megaspore to embryo sac, there were no reduction in the number of chromosomes. For that case, A, which is antipodal, will contain the half number of chromosomes of the parent cell, which is 11. It means the antipodal cell will have 11 number of chromosomes. The egg cell, normally the sexually producing organism contains the gamete cell having haploid number of chromosomes. Whenever you see the gametes has the haploid number of chromosomes. If the haploid number, the egg cell, were produced when the megaspore transformed in a series of mitotic division 3 times, it did not reduce the number of chromosomes. It remained with the same 11 number of chromosomes because it is a gamete cell. C, the megaspore mother cell. Remember here, the megaspore mother cell contains the diploid number of chromosomes of the parent cell, and it's the one that undergo meiosis. If initially it has the diploid number of chromosomes, it means the answer for that question will be 22 number of chromosome. But you have D, the polar nuclei. I'm not going to answer this question. I leave it to you to examine what will be the number of chromosome in the polar nuclei that appear to the center of the embryo sac. By so doing, you will have answered this question correctly. What you need to understand, or what you need to remember, is the role of meiosis and the role of mitosis will make you comfortable to respond to this question. Dear students, I have managed to show how question number 1 to question number 5 can be answered. What is very important is to make revision in all aspects I have presented to you here. What to remind you again is to make revision so that different concepts I have been presenting here can stick in your mind. Before I wind my presentation, I would like to extend my sincere thanks to you all students who have been listening from the start of this presentation up to the end of this presentation. I say thank you for listening. Goodbye.